So I would like to go back to a couple of slides about the basic behavior properties of the musculotendinous unit, and we'll do this quickly, and then I'll move forward to the muscle functions as well as the force, velocity, and length tension. So we remember we talked about this, that muscles are the organ in the body that produces force, correct? And because of the structure of our skeleton, all our movement are angular movement, right? So what is the quantity that we should be measuring, and most of the time for the muscle activity? It's not going to be linear force, correct? So that quantity is going to be moment or torque, because we have tendency to rotate in the muscle, in the skeleton, because of the muscle. And so the torque, remember the torque, what is the function formula of the torque? The moment or the torque equals to what? Force times the perpendicular distance, after distance. And we're going to have a couple of exercises along the way of measuring the torques generated by different forces. Now, we are going to be focusing on the knee, right? Because ACL tears, that's our target this semester. So before we go there, we talked about something called fast twitching and slow twitching fiber. I would like to go back to it and talk a little bit more. Now, fast twitching fibers, I need to describe a couple of things for that. For fast twitching fiber, it reaches peak tension quickly and relax also quickly. However, the slow twitching fibers, they take more time and they stay for a longer period of time duration. Now, here's my question to you. Which one do you think uses ATP? Fast twitch uses ATP as well as also the slow twitch. So both of them need, why do you think we need ATPs? What is the thing that is going to use ATP? What's the molecule that will use ATP? It's the myosin, right? You remember the heads, movement, the head? So the myosin heads are the ones going to be moving. So these myosin heads are the one that needs ATP. Now, here's the thing. Fast twitching and slow twitch. Fast twitching does not, so it needs ATP, but it is anaerobic reaction. Do you know what does that mean? So it is using glucose, right? Glycogen, instead of oxygen to generate ATP. So our muscles, they do save some, glucose products. So that's the fast twitching. It does not need to have oxygen. Now, here's a problem with it. That means that it is not sustainable, correct? Because we need oxygen. Now, the slow twitching fibers, they do need oxygen. And because of the oxidative process of creating the ATPs, it stays for a longer period of time. So that's sustainable. Does it make sense? Now, how does oxygen get into muscle? What is the molecule that holds, have oxygen? Hemoglobin. Now, there is something in muscles do the same function. Do you know what that is? Myoglobin. Now, myoglobin is red, right, in its shape. So that means that the fast twitching, do they need oxygen? No. So they don't have any of the myoglobin, correct? Now, what do you think the color of the fast twitch will be? White. Because it does not have any myoglobin, right, or blood cells. We don't need those. Now, this slow twitching, they will be red in its, color. Now, here's the thing. Do you think that we have muscles that are either fast twitch or slow twitch? Or do you think they're going to be all mixed up, the fiber? All mixed up. All the fibers are mixed up. And that's why you have the color of the muscle that is actually reddish or close to pink. Does that make sense? Now, that's important because the fast twitching does not need oxygen to do the contraction. And that's what I wanted to, cover in the next slide when we move there. So we have type 1, the slow twitch, and then I have two types of fast twitching fibers. This type 1 slow twitching, contraction speed is slow, fatigue rate is slow, diameter is small, right? ATP concentration is low because we are using oxygen to create the ATPs from the mitochondria, right? In the cycle, the Krebs cycle, high. And then we have glycolytic enzyme concentration is low. That's glycolytic enzyme is the one that is creating the anaerobic ATPs. Does that make sense? How you look at that? Now, we have two types of the fast twitching. We have fast twitching that needs oxygen, and fast twitching fibers, they don't need oxygen. They use ATP, they produce ATP from the glycolytic enzymes, right? Now, that's fast. The fatigue rate is intermediate. Fatigue rate is fast because you are depending only on the glycolytic enzyme to do the ATP for creating the ATPs. This one, it uses oxygen. That's oxidative. So fast twitching, we have two types. One of them use oxidative, and the other one is using glycolytic enzymes to create ATPs. Now, the diameter is large for fast twitching fiber because we need a lot of myosin and actin in doing this. Just make sure don't spill it on your laptop. Mitochondria. If you are using oxygen, we have high mitochondria concentration. If we're not using oxygen, we don't have much. A glycolytic enzyme, it's low in the type 1 because it's slow twitching. It needs oxygen, but it's high on type 2. Does that make sense? Now, engineers. We are engineers. How would you assess how much fast twitching or slow twitching fibers in our muscle non-invasively? These are the things that we would like to think about. Well, MRI, because, and you can look at which one is larger, which is smaller. That could be, but expensive on the other hand, right? You can look at the color of the muscles, but that's also invasive. You can look at oxygen concentration, and that's an MRI actually. Okay, it looks at the water concentration, which is an indication of how much oxygen do you have. And also you can track red blood cells, hemoglobin, or myoglobin in those. Okay, which kind of exercise do you think you would need the fast twitching in? Sprinting, right? Because you need speed, correct? But if you want something durable, then you need the slow twitching fibers to activate. Now, going back to technology, it is still big question. People do not have a gold standard approach to assess the concentration or the number of fast twitching fibers compared to slow twitching fibers. So this is still a technological problem for us. I like to look at EMG. So here's the thing. Let me tell you something. If you are doing fast twitching, what would you expect to happen with the EMG signal? So what does fast twitching means? That you have a lot of contraction, right? And it's a big diameter, which means there's a lot of ions going in and out, which means you have higher frequency of this zigzagging of the EMG, as well as a higher amplitude, right? Because that's going to be correlated to contraction. So one of the experiments that we can do is actually we can bring someone and have some dumbbells, and we keep increasing the load, and then do the EMG for the biceps, right? And then you can correlate how much load is lifted and how fast with your EMG signal. So we can use surface EMG actually to assess the fast twitching or slow twitching recruitment, or fast twitching and slow twitching fibers recruitment. And that's another experiment actually we can do. But we need to find, some kind of motor tasks to do that. This is what and that's one. So we talked about the size MRI. We talked about oxygen concentration. We talked about the color. And now you can talk about the relationship between, firing of the EMG and the load generated. So these are the things that as engineers we need to think about. Make sense? We go with that? Yeah, so I wanted to make sure that we are on the same page about which one uses oxygen to generate ATPs and which one is not using oxygen to generate ATPs and using the enzymes. Okay, slow twitching motor units. What is the definition of a motor unit? So slow twitching motor units are easier to activate than fast twitching because we have oxygen that's sustainable. ATP concentration is consistent. Now, slow twitching motor units are the first to be activated because they have the oxygen. As the force requirements, speed requirements of the duration activity increase, motor units with high threshold are progressively activated, and that's something. Now, thus slow twitch, yeah, so we're good. Now, here's the thing. I would like to go back to concentric and eccentric and isometric concentration one more time, just to make sure that we are on the same page. Now, if you are lifting weight this way, right? Lifting weight up, lifting up. What do you think is happening? Is it concentric or eccentric? It's concentric because you're going up and you are pushing it. You're doing contraction on the triceps, right? So it shortens to get you up there. Now, let's do it this way. Eccentric means that you are doing load, but you are also elongating, correct? So let's say I'm holding, how much do you think I can hold with my hand? 50 pounds max? That's too much, right? If you have a very heavy load and you're trying to push up, but it's going down, what is that? So it's very, very heavy and I can't hold it up. So I'm trying, but it's taking my hand down. That's eccentric. If you can control it, that's concentric. Because the elongation, while you are trying, it's pushing, it's going down. That's eccentric contraction. Any contraction with the length of the muscle, that is isometric. Flexion, right? That's concentric. Easiest thing to think about. But eccentric is tricky because you need to make sure that you are eccentric. You're losing control. It's elongated. It's elongating as you're trying to push, but you're failing. That's eccentric. Now, pulling up is concentric. Here's the thing. If you what do you call when you are hanging from with the bar and you go up and down? Pull-ups. But when you go down, that is eccentric. When you go up, eccentric, you're still trying to push it, but it's going down against your will. When you go up, in the bars, when you go down, you want to go down. That's controlled, right? But it's elongated. It's getting elongation. So the weight is pulling you down, and you're trying, to keep hold onto the bars, and that's eccentric. See, let's say this is 200 pounds, right? But the movement of the arm is going down because you can't hold it. It's pushing, it's taking your arm down. That's eccentric. And now you need to think about all the muscles that you use during any activity and see which one is concentric and eccentric contraction. That's important. And then, hopefully this is clear. And then we talked about the agonist and antagonist. The agonist is the muscle that causes the movement. Antagonist is the one that slows the movement or relax or elongated. So one is shortening and elongated. Now, stabilizers. Let me give you an example. I wanted to go back to this one because that's important. If I am doing this, if I'm flexing my elbow, like flex elbow, right? Correct? The biceps is actually the agonist, correct? And the triceps is the antagonist. Now, the stabilizer is a muscle that acts to make sure that the movement is happening proper. For example, what do you call this muscle here? Deltoid? No, right? No, the deltoid. Now, in the inflection, the deltoid inflection is actually your stabilizer. Because if you don't have the deltoid doing this, actually when you try to pull it up, it's going up, it's go up. So there's something stabilizing your movement so that you can have the upper arm stable and to do the flexion. Does that make sense? So you will have other muscles that work with the agonist and antagonist in order to stabilize the movement. That's the point. Now, neutralizer, also they eliminate any unwanted action. So if something is going to cause when you do flexion and something is moving your elbow in the frontal plane, right? So that's called a neutralizer. Now, we need to understand this because as if you noticed as we keep moving, there's a lot of things that we can measure and assess using EMG. Now, you need to know which muscles you need to always track. The easiest thing is to track an agonist and an antagonist first, but sometimes it's the stabilizers and the neutralizers are the important one, especially for ACL tears. Does it make sense? Because the tear will happen when you have unwanted or when you are something is moving against what you wanted to. So if we're doing research together, we'd like to look at EMG signal, not just the force plate. Now, for EMG, for the muscles, we need to identify which one is the agonist, antagonist, and which one is stabilizer and neutralizer, and then take signal from all of them. Make sense? Now, for knee flexion, what is the knee flexion? How would you do the flexion? That's flexion, right? Correct? Now, what is your agonist and what is your antagonist? In your flexion, knee flexion, and then the other one is the quadriceps. That is going to be elongating, right? So that's inflection. And when we did the jumping in the experiment, we used those actually as agonist and antagonist. Does that make sense? So if you would like to assess EMG for the muscle, you need to assess those. What is the quantity that we're going to get from this? How much force is produced? Fast twitching, slow twitching fibers, right? Correct? We can get that from the EMG. We can look at the EMG. But if you need to look at the tendency to rotate, you don't need the agonist and the antagonist. Which one do you need actually to assess? The stabilizers and the neutralizers. Does it make sense? So to us, it's not just I know that it's anatomy that you covered, but when we look at a joint, for example, the knee, you need to know these things, not just the internal structure of. Okay, let's do an exercise. We will start with this. We have 3 pictures. I'm going to do it twice. So let's go into 3 groups. And what I want from you in these experiments, if you can see this, I don't know first of all, describe what does that, you need to tell me what that movement is, and tell me what is the agonist muscle and the antagonist muscle. And let's go into 3 groups, and let's do this for the first one exercise, 7 minutes? Yeah, this year. We're going to do the other ones, but we need to understand which one is the agonist and which one is the antagonist. So a quick review of our muscle anatomy. First of all, describe what is the movement. Yeah, you're mostly athletes, so you know probably what is that. So I think it's pulling the legs back, back. Maybe suggest. But let's focus on agonist and antagonist. Maybe stabilize. See which one is going to cause. You do. Yeah, I need the agonist and antagonist. Because I don't know the other ones, myself. Okay, we'll move to the next one. Next one. We good with this one? Okay, next one. These things. What are they called? what is the agonist and the antagonist, the main agonist and the main antagonist, and what do you call them? Yeah, this is important because when you try to measure EMG, you need to make sure that you always measure agonist and antagonist pairs. Make sure that you write those, and we're going to upload this onto a Dropbox, right? I'll give you 2 more minutes, and we'll upload those to a Dropbox. Yeah, this movement has to do something with the shoulder, with the back, with the muscles of the on the back. Yeah, and this is really important. You always need to think about any motor task, which one is the agonist, which one is the antagonist. Now, here's my question to you. So when we did the cross in the lab, yeah, the cross movement, which one do you think is the agonist and the antagonist? Because we went in an angle, we went that way. So you need to look at the knee and think about which one is going to be your agonist and which one will be the antagonist in the knee. That's an exercise to think about because we're going to be doing research on this. But that's important. Which one is agonist and antagonist when we do that movement? This could be a fun exercise actually to the coaches, to take it to the coach and tell him, what do you think? Which one is which? Okay, we good? Complete Anatomy, the software, I think they do have something about these. They can tell you exactly which one is which and what's happening. The Complete Anatomy. Okay, if you got confused about any of these, you can ask your coaches or anyone, and because I'm going to be searching for that myself. I don't know exactly all of them, but I know some of them. But I would like to, and I will ask you about the stabilizers and the neutralizers, the other muscles. Okay, so we're good with this. And that's a good exercise. Now, yeah, this is another thing I wanted to go back to from last time. Here's the thing. We have something called active and passive insufficiencies. And this is related to muscles that passes multiple joints. Okay? Now, let me show you something. If I did this, right? So what do you call these muscles here? It's not their names. It's a group as extenders. Yeah, extenders. But I would like to discuss one more thing. The passive insufficiency is related to exceeding stretching a muscle to a maximum length passively, right? Extensors, those are the extensors, right? These here, the muscles. If you did this and you try to close the fingers, it's not going to work. Because we have stretched these extensors all the way, and remember they are passing through 2 joints, this joint and that joint. So that's a problem here. Now, on the other hand, when we say active insufficiency, if I did this right this way and I tried to close my arm all the way, that's active insufficiency. Let me show you something. I want to draw something here because that's important. Do you remember the myosin and actin filaments? Right. If you have your hand this way, this way, it means that your actin and myosin filaments are actually like this. You'll have this one, you'll have that one, and you'll have this one, that one. Those are my actin, right? And then you have this myosin filament, right, here, and you have the arms both sides, correct? Now, if you are contracting, if the length is, if the actin filament are not overlapping, what do I mean by overlapping? If you are like this, look at that. Okay, see these two actin filaments, this one and that one, they are overlapping on top of each other. Do you know what does that mean? If you look at the muscles here, when you squeeze your hand this way, you are bringing the myosin, the actin filaments all together so that they are overlapping. So the myosin does not have much places to connect to, the heads of the myosin. And that's why, which means that this is active, which means active means that you already contracted the myosin and the actin to its very, very short space. And now if you try to do another contraction, it's not going to happen. That's called active insufficiency. Does that make sense? The passive insufficiency, that you stretch the length to its limit. But active insufficiency, it means that your actin filaments are now overlapping and the myosin does not have places to connect to. And that's called active insufficiency because there is no room for them to contract anymore. So what are they going to do? They can't. You already contract the sarcomere, right? So it's already stretched inward, squeezed. Does that make sense? So if it's this way, they're relaxed, you're not stretching them, you can open and close your hand. But if you did this for the extensors and try to close, that's going to be passive insufficiency. Now for the muscles of the hand itself, if you do this and you try to close it, it's active insufficiency from this side. So one from this side and the other one from this side. Yeah, I wanted to make sure that that's clear because we didn't talk about this last time. It was quick. We did it quickly. Are we good? anytime you have your actin filament overlapping on top of each other, that's active. Something is acting, and pushing it inward, which means that we don't have much space to contract. There'll be small force and small spaces to contract. So passive insufficiency, inability of a two joint muscle. So these extensors, two joint muscle to stretch enough to allow full range of motion, and that's what I explained, right, from the extensors. And if you have something called active insufficiency, it's a condition when two joint muscle cannot shorten enough. So when it's almost already shortened, we can't shorten it more. That's called active insufficiency. And so the two muscles cannot shorten enough to cause full range of motion of joint it crosses at the same time. We good with these two? Now, for us, this is a test that you can use also the range of motion and EMG to find the health of these muscles, because those muscles they're connected to two joints. Those are important. They're very important muscles. And also the hamstring and the quad are also multiple joints actually, because it's the hip and the knee. So these are the things that we need to make sure that when we talk about the range of motion, that we can assess passive insufficiency and active insufficiency. I'm a big fan of EMG, and EMG is a good assessment tool for these passive insufficiencies and active insufficiencies. Okay, can you think of a knee insufficiency? How would you imagine then? Because we are interested in ACL tears. So what do you call this? Let me show you this. All right, are we good with this? Passive and active. Now, here's the thing. We care about the knee. Always think about the knee. What are the things that we can do for the ACL tears? Okay, that's done. Now, going to the force, velocity, length, tension, electromechanical delay. That's another important action. We talked about the concentric and eccentric, right? Concentric, let's say you have a weight and you are pulling it up, right? That's concentric. Now, here's what happened. The larger the weight, the slower the velocity, correct? You can't push it fast enough. Eccentric, it's the other hand. If you are, on a bar and you're pulling yourself down, the larger the weight, the faster the velocity, right? The larger the extension, the go down faster for that. And I told you that you need to make sure that as an athlete, you need to know your isometric maximum load that you can hold, and then you can study the concentric and the eccentric. And that's force-velocity relationship of muscle tissue. Now, where is this thing important? If you are doing, you're athletes, a lot of you athletes are. What is it that's important to you, speed or force? It's velocity. You need to do things as quickly as possible in most sports, in the majority of them. But if you are, let's say, lifting weights, right, you need force. That's for sure. So yeah, it depends, but usually people want to do things as fast as, even lifting weight, you want to do it as fast as possible so that you don't damage any of the tissues. Okay, so the force-velocity relationship of a muscle tissue, when resistance or force is negligible, force contracts with maximum velocity, and so as the load increases, concentric contraction velocity slow down to 0. We understand that. As the load increase, eccentric velocity increase. So if you are on a bar and you're going up and down and you have weight, the larger the load, the faster you go down. The heavier the subject, the faster they go down. That's eccentric. Remember, we're trying to pull ourselves up, but it's pushing us down against our will to go up, and that's eccentric contraction. So the larger the load, larger velocity, that's eccentric. Larger load, slower velocity, that's concentric contraction, with how you look at those. It will be also, again, a cool experiment to look at the EMG activity as well as the ranges of motion when you do these eccentric and concentric contractions for them. Okay, now for the length tension relationship of a sarcomere, that's the thing that I was mentioning to you. So here's the thing. When we say passive tension and active tension, the total tension in a sarcomere presented in a stretched muscle is the sum of an active tension and the passive tension. Now, here's what happened. Let's work with the passive tension. The passive tension is that when you are stretching a rubber band, what happened to the rubber band? What is it doing to you? Just a rubber band. It's going to, your hands will feel stronger forces, right? As you stretch it, you feel forces trying to pull back. And remember when we talked, we have elastic components in the muscles. So if you stretch them, especially the tendons, if you stretch them, they'll try to go back. So they're creating force. That is what we meant by passive tension. See this? as you increase the length, so the x-axis is length, as you increase the length, the force is going up. Do we agree with this? As you stretch it, it's trying to go back to where it should be. Now, for active, now the active tension is the one that's this one. This is the line with the dashed line. That's called active tension. Forget this one that's going up. That's the addition of the two. But the active tension is this one, this line. Now, active tension is the myosin and actin filament going together, correct? Now, let's look at this point here. If I'm at this point, this means that we stretch the muscle all the way, because that's length on the x-axis. So at this point, my muscle is stretched all the way, correct? Now, why do you think it's low tension? It's creating a very low tension. Because if you have actin this way and another actin here, the myosin will be like this, right? Because you stretch the actin all the way back, away from it, sarcomere, correct? So the heads of the myosin is barely touching one actually side of the actins. Does that make sense? So they're far away from each other. That's why the tension is low. We don't have anything to pull. The tension generating molecule is the myosin heads, right? The myosin heads. Now, what happened when we are here? When we are here, you have the actin like this, and you have all the myosin heads connected to the actin, right? So all the heads are connected, and I'm generating the maximum force and tension. Does that make sense, right? Now, what happened when we are here at this point? So what happened here? I have the shortest length of the muscle. So remember when we discussed the active insufficiency? We said they're very close to each other. So the actin are on top of each other. The actin filaments are on top of each other, and the myosin has nothing to connect to. That's why the tension is going down, right? So here, at this point, the actin filaments are overlapping. So the myosin has a very little space to move, actually, even if it can move, actually, the actin. And that's why the tension is low. Now get this one. If you add the active, right, with the passive. So here's the fun part. Until this point, you can stretch your muscle. Look at this. You can stretch your muscles until this point without having any passive tension, right? And then when you start stretching it more, then you'll have passive tension, and you can start adding the load here. And that's why the end figure of your tension is going to be this one, right? So for different muscles, as an engineer, you need to plot this figure, identify this point, and see how much tension you can do. Another important aspect. What if you kept stretching the muscle more and more? What's going to happen to this curve? Keep going up until it ruptures. Can you think of an example that this is a dangerous thing for us? ACL tears, it's one of them. But do you think can you imagine the structure? So how would you? Let me give you another example. Do you know if you're doing what is that flexion your bicep curls standing in a 90 degree, sitting on a 90 degrees? What if I did this? So what I'm doing with my biceps, I'm stretching them. And you know they have chairs that they are tilted 135 degrees, and you can go back, lean back, and then start doing the curls. That's very dangerous. That's extremely dangerous because you stretched your muscle and your tension now extremely high now, and that's when you actually damage your ligaments and tendons and the muscles themselves. Does that make sense? Stretching the muscle going to increase your tension, and that's something that people actually do not look at when they do exercises. Even coaches, they need to know this. If you stretch the muscle and start doing contractions, you are in this region. And that's going to damage the muscles or the tendons and ligaments inside it. So whatever activity you do, you need to think about this. Am I elongating the muscle before doing the exercise, or is it in its relaxed part? Because that passive tension, that is the killer. Alyssa, that is what I think is actually the problem, is the passive tension that can actually result in ACL tears. And that's another thing to think about. What is the thing that we are stretching to create that passive tension of the ligaments and tendons? Make sense? We good? Alrighty. Again, as engineers, we love numbers, we love figures, but we would like to see what does that mean in the real life, in the activities of real life. Okay, what else? So here, factors affecting muscle force generation, strength shortening cycle. So this is another thing actually to look at when we do exercises, and I like this because it's related to the knee as well. Stretch shortening cycles. It's eccentric contraction, in which the muscle is actively stretched, allowed immediate followed immediately by concentric contraction. And can you think of examples? This is an example, right? You go down and then you jump. See, you are stretching the muscles here. You have eccentric contraction, elongation and contraction happening there. That's also muscle fibers, go stretch, shortening, stretch, and shortening, and that's what happened to the muscle, right? There are a lot of things to do, but I always like to think, what is the technological thing that I can measure or assess, or a tool that I can build? That's the key thing. So the difference between us and exercise science people or physical therapy, that we would like to build things and to assess, using the technology. Okay, one more thing, and I will stop after that. So factors affecting muscle force generation, we have something called electromechanical delay. We agree that the trigger for contraction is an electrical signal coming from the brain, right? The action potential from the brains are the ones that are going to trigger the contraction of the muscles. Now, usually we have a delay between the EMG and the force. When the EMG is active, so if you have sensors and you're doing, let's say, contraction, the EMG will appear, but then the force itself, the generation force, will take a delay. See, between EMG and force? And I would like always to plot EMG and force generation at the same plot to look at that delay. And that is called the time between the arrival of neural stimulus and the tension development by the muscle of an external object, called the electromechanical delay. Make sense? We good with that? I'll look at that. Yeah. The other slides, we're going to continue with them, but I wanted to stop here because that is a good point to stop. And the good thing is that I will be able to. Yeah. I thought that a couple of things last time we did not cover thoroughly. So now I'm happy. I'm happy that we have that thing. We good? Okay, so next time I will be doing these, the torque. Yes, that's going to be an exercise. Yeah, I want you to upload that to the Dropbox. We have week 7 Dropbox still there. Are we in week 7 or week 8?